Okay. It's always good to have a question in the title, although in this case it's a bit difficult title. In fact, I think although it's quite easy because I just have to answer yes or no, and I think I have a good answer for this. Can we stimulate the bone formation, and can we actually generate the machinery to promote new bone? That is something you can see that my research group here in the background is also thinking about when they're preparing a tissue from our animal study. The bone formation is a highly regulated process. It involves several factors, and it has been presented as a diamond concept, including mesenchymal stem cells, cell mediators, cytokines, and the scaffold, and the mechanical environment. But this has been even enlarged to a little broader concept involving still the mechanical environment, which usually the surgeons are thinking of, and also the osteoconductive scaffold. And as we know, the vasculature is very important. That's why we also quite often bring the vascularization to place, for example, with flaps or something. But we also have these other factors we should think of: the inflammatory cells, the osteogenic cells, and the growth factors. And if we want to have a good fracture healing and use materials, we must think of the whole concept, so we need answers to all. The mechanical environment, I'm not going to talk so much about that. I'm going to talk more about the other more difficult factors. We quite often use the induced membrane technique when we have difficult fractures and situations, and it has become quite popular. If you think about the technique, which involves actually a PMMA spacer that could, and we think will act as a spacer that promotes a membrane around itself, and then after a certain time we can remove the spacer and put in, for example, autograft bone or a bone substitute, and we do it in a two-stage procedure. So the interest of the induced membrane has raised in popularity, and we are thinking about that. But if you have a membrane, the thought is, of course, that it will do something. But does it really do? And what and when? The fact that there is a membrane is not, in fact, always the answer. Because I work as a hand surgeon, and if I need something to protect my tendons, if I don't have a tendon sheath, I just put a silicone spacer, I wait a few weeks, and then I have a tunnel for my tendons, and I have a membrane, but that membrane does not do anything at all for this. So that you have a membrane that doesn't actually tell that the membrane is doing something, and that's why we need to do research on this. And our preclinical research is based on the membrane technique, because we want to actually know what happens in the membrane and if we can actually do something with the membrane so that we could get better results. We know that it hinders absorption, we know that it conveys vascularization, it contains osteoprogenitor cells, and it can release, but we have not studied it as much as we probably should. There are also questions about this technique. You need 2 invasive surgical procedures. We are thinking of if you use PMMA, which is the standard, could we use it in a single stage? That we already know. But what is exactly the evidence of this membrane? So by really understanding the induced membrane, I think it's a very good option to really come closer to the biology in bone regeneration, and when we understand the biology, we can start manipulating it also. So the purpose of our study has been during several years now to develop a bioactive scaffold which would be suitable for a one-stage procedure, and the end what we want to achieve is a bone substitute for a defect defect, because I think we don't have tools enough for this region, and this is the most difficult region to treat. We are doing this in tight collaboration with Åbo Akademi University, which is the university that provides us with the glass. And the glass we are using, we have several which we have tested, and we are doing this really thoroughly in a systematic way, and we have chosen to present here the sintered S53P4 granule glass, which sintered means that you have put the granules together and then you get a block. And we also had tested if we could actually coat this material with the PLGA polymer, so that we have the thought that we could actually do a small inflammation that could trigger the biology around this implant. So we have a study on 36 rabbits, and we did it in the beginning on the metaphyseal area, because we wanted to start actually from the beginning. And we made control drill holes as a control, because if you do a trauma, you will also have these triggering factors, we have the inflammation and so on. So we need to have one control drill without anything. Then we put PMMA rods, and we had also bioactive glass rods, and then bioactive glass rods coated with the polymer. And this is the operative technique. We put these rods, which are 5 millimeters in diameter, in here. And here we left it open, so the glass is in contact with outside, and we get the induced membrane, and we have analyzed this, and we had also naturally analyzed the bone formation here. And what we saw was that it works very good as an osteoconductive scaffold. Here is the glass, and here is the PLGA-coated glass, and you can see that in both cases we can see that it's an osteoconductive material. Then we looked at the vascularity, and we took from the membrane histology from all these, and then we also did PCR VEGF expression studies, and it was really interesting, because in the beginning, if you look at the control, you see that you get an input here, but it diminishes after time. Here is 8 weeks, 4 and 2 weeks. In the beginning, all these materials are quite active. Here you see that they are not so active, but what is interesting is that we saw that the polymer has a small delay when the polymer degrades, and you see an increase in this capillary account here, which is also seen as the VEGF expression. So this was a very good result. We can see that we can actually manipulate the bone substitute to have an increase in VEGF expression as well as capillary count. And we looked at also at the inflammatory response, and we saw a little bit at the same where the polymer-coated bone substitute had a little more influence on that. Okay, but how about the bone formation? Do we see anything that we can compare the vascularity and the bone formation with? And we looked at the BMP 2, 4, and 7, and PCR results from that. And just to remind you that PM2, the role of PM2 is in osteoblastic differentiation. It should be more active in the beginning of the bone formation process, and these two should be in the middle or in the end of it. So if you look at BMP2, you would like to see something here in the beginning. We saw that all these have impact on the bone formation or these factors. And if you look at the end here, we also saw that there are some differences, and again we could see that the polymer-coated glass had impact also in the end when we want to, although it's a little bit hindered in the beginning as it degrades. So how does it look actually on CT? We took CT from the cortical, middle and inner side of this tube, and all these 2, 4 and 8 weeks. And when we looked at that, we could see that it will grow so that there will be bone formation more and more. And we have statistical significant differences between this in the polymer, that it is a little bit slow in the beginning, but it has positive effects on other things, and then it comes up in the end. And in this we could also see that it was a little bit better than the glass itself. Then we, as the last one, looked at the osteogenic cells and looked at how does these interfere with our two scaffolds. So we put mesenchymal stem cells together with the scaffolds, and we put them 11 days in a media, or two different media, an osteogenic medium and a basal medium. And the red one, which you will see here on these pictures, is allicin red, which indicate intracellular calcium. And this was quite interesting because this is the positive control, and we can see that if you look at no scaffold, this is the control, we don't see anything here. But if we put only bioactive glass here, and in the negative control, where there should not be anything, according to this, we can say that glass is 53P4 is very osteogenic. And on the contrary, we saw that this polymer does not have the same effect. So the conclusion is that the induced membrane appeared to be similar or superior the technique compared to PMMA-induced membrane. When we're looking at the bioactive glass, we get the same results. The S53P4 glass has a stronger impact on bone formation than the polymer, especially in the beginning of the implantation, but as the polymer a little bit hinders it in the beginning, but that is catched up in the later. And if we look at the vascularity, we see that the polymer-coated glass has a little bit stronger impact on the vascularity than the glass itself due to the inflammatory response caused by the polymer. So by combining these kind of osteoconductive, osteostimulative, angiogenic effects with these PMMA and VEGF-induced PLT coatings, we are now heading for a scaffold for a possible single-stage EM technique for diaphyseal defects. And the answer to this question is therefore yes, we can manipulate the biology, although it's a little bit complicated. And this is the first picture of a sintered bioactive glass without the polymer that has been implanted in rabbit tibia, and you can see that it's quite nicely attached there. We don't know yet how it looks like here inside. These are the very few first pictures of this, and we are now analyzing the induced membrane and all the other factors I showed you from the metaphyseal area. Thank you.